The cell membrane, also known as the plasma membrane, is a critical structure found in all living cells. It is composed primarily of a phospholipid bilayer with embedded proteins, and it serves several essential functions. Most importantly, cell transport. Cell transport is the process of moving substances across the cell membrane, which is critical for maintaining homeostasis. Throughout this video, we will discuss different types of cell transport, but before that, we have important vocabulary to define.
Starting with the lipid bilayer. The lipid bilayer is the main component of the cell membrane. It's composed of two, hence the root bi, layers of phospholipids. Phospholipids are the main molecule of the membrane. Each has a phosphate head, which is hydrophilic, meaning a molecule attracted to water. The inward part of the lipid bilayer is the fatty acid tails, which are hydrophobic, molecules not attracted to water. Together, hydrolipids are amphibiotic, meaning they have both hydrophilic and hydrophobic parts. Cholesterol is also found scattered throughout the inner part of the cell membrane in order to keep the membrane fluid, not too rigid or too leaky.
It is also important to note the difference between unsaturated versus saturated fatty acids. Saturated fatty acids are more tightly packed due to the straight chains, while unsaturated fatty acids are more loose due to the more kinked chains as a result of double bonds. Saturated fatty acids will be more rigid, whereas unsaturated fatty acids will be more flexible. Cell membranes oftentimes have both saturated and unsaturated fatty acids to balance flexibility and rigidness.
Now, the most essential aspect of the cell membrane in regards to cell transport would be membrane proteins. There are two different types of membrane proteins, one being peripheral proteins. These proteins sit on the inside or outside surface of the membrane and are primarily used for cell signaling, which is the proteins acting as receptors for extracellular signals, such as hormones and neurotransmitters. These proteins are also used to maintain cell shape. The other type of membrane protein are integral proteins, represented by these five structures. These proteins are primarily used for transport of molecules throughout the cell membrane. Membranes also include cell markers, which are glycoproteins and glycolipids. Glycoproteins are proteins with attached carbohydrates, and glycolipids are lipids with attached carbohydrates. They are involved in cell recognition, signaling, and adhesion. Glycoproteins are used for cell signaling, whereas glycolipids work as cell recognition. It's important to remember glycolipids are attached to the fatty acid, the phospholipid tail.
Now that we've gone over all the important vocabulary, let's explore different types of cell transport. Before we go into detail, it's important to understand different concepts. Concentration gradient is the difference in the concentration of a substance between two areas, which drives the movement of that substance from a region of higher concentration to a region of lower concentration. When molecules are moving with the concentration gradient, high to low, they don't require energy. This is known as passive transport. When they are moving against the concentration gradient, low to high, they require energy. This is known as active transport. For example, these oxygen molecules flowing freely through the bilayer represent passive transport. Pay attention to how there's more oxygen molecules on the outside than the inside. Thus, following the concentration gradient, they diffuse through the cell membrane into the cell. Small, nonpolar molecules usually go through this transport, such as nitrogen, oxygen, and carbon dioxide. Occasionally, small, uncharged polar molecules go through, such as water, but not as effectively. The example shown can also represent simple diffusion.
Next type of cell transport will be osmosis. Osmosis is the diffusion of water through aquaporins, or the bilayer. Pay attention to the example shown. Here we have an aquaporin pump. This is a membrane protein. More importantly, an integral protein used for transportation of water. This is passive transport, because water passes through these pumps without any ATP energy necessary. Fun fact, a single aquaporin can transport up to 3 billion water molecules per second. Aquaporins are highly selective. They prevent the passage of charged particles and other molecules, ensuring only water.
Next, we will discuss facilitated diffusion, which is also passive transport. Facilitated diffusion is where molecules move down from the concentration gradient from high to low concentration with the help of membrane proteins, but without using energy. For this example, pay attention to the leak potassium channel. This channel helps potassium ions pass through the membrane in order to maintain homeostasis.
There's multiple different types of facilitated diffusion. We'll go over two more. The next one being the gated potassium channel. This channel opens in response to a signal, like a voltage change or ligand binding, allowing K+ ions to flow down their concentration gradient. They typically move out of the cell since there's usually more K+ ions inside than outside. The final one being the GLUT carrier protein. This channel moves glucose across the cell membrane down its concentration gradient.
Next, we'll discuss active transport, which can be represented by the sodium-potassium pump. This pump utilizes energy, making it active transport. 3 sodium ions go out of the cell, where 2 potassium ions are moved into the cell. This is used when the sodium concentration in the cell is too high.
Finally, we'll discuss endocytosis and exocytosis. Both of these processes require ATP, which is energy, making them active transport. For the sake of representing these transports, we're going to remove everything that represented the earlier vocabulary from the cell membrane and just show the cell membrane. But do remember, all of those things are present. Endocytosis is when the cell membrane forms a vesicle to bring in wanted molecules or ions inside the cell, represented like this. Exocytosis fuses with the membranes to send unwanted molecules or ion out of the cell.